They ran the tests. They compared the data. They looked at two of the most dominant human populations on the planet, Chinese and European, side by side, genome by genome, chromosome by chromosome. And when the results came back, the scientists didn't celebrate. They went quiet. Because what they found didn't just rewrite a chapter of human history. It rewrote the entire book. Two populations, one planet, separated by thousands of miles and thousands of years. And yet, the DNA told a story that nobody expected, nobody predicted, and almost nobody is talking about. Until now. Here's something that almost no mainstream outlet has explained to you properly. Scientists have spent decades building the foundation of genetic research almost entirely on one group of people, Europeans. The overwhelming majority of early genome-wide association studies, the foundational databases, the landmark papers that shaped modern medicine and our understanding of human biology, they were built using European DNA as the baseline, as the default, as the standard against which all of humanity was quietly measured. Think about what that means. For decades, when a doctor prescribed a drug, when a medical algorithm calculated your disease risk, when a genetic test told you something about your body, it was speaking in a language written almost entirely for one population. In the rest of the world, billions and billions of people were essentially receiving answers to questions that were never asked with them in mind. And then scientists started comparing, seriously comparing, Chinese DNA against European DNA. East Asian genomes laid side by side with Western ones, studied not to confirm what we thought we knew, but to discover what we had been missing. The results did not just surprise the researchers. They fundamentally challenged assumptions that had been embedded in science, medicine, and our understanding of what it means to be human. Here is where it starts to get deeply uncomfortable. Asian populations account for more worldwide genetic diversity than all of Europe put together. Read that again slowly, because it goes against everything the Western-centric model of genetics has implied for the past half-century. Europe was treated as the template. Europe was treated as the most studied, most understood, most representative population in human genetic science. And yet, the data, when finally examined honestly, shows that the genetic landscape of Asia, and of the Han Chinese in particular, contains layers of diversity, variation, and complexity that Europe simply does not. A study published in the prestigious scientific journal Nature reported findings from over 1,700 Asian individuals, demonstrating at least 10 deep lineages that produced the various ancient Asian populations, with the equivalent of just one for all of Europe. 10 deep lineages against one. That is not a minor statistical variation, that is a structural revelation. That tells you that the human story did not begin and radiate outward from a simple point. It tells you that the roots of humanity are far older, far more complex, and far more deeply buried in the East than our dominant scientific narrative has ever comfortably acknowledged. But here is where it gets even more layered. The Han Chinese are not simply one group. They never were. The Han Chinese are the largest ethnic group in the world, and their origins, development, and expansion are complex. Many genetic studies have shown that Han Chinese can be divided into two distinct groups, Northern Han Chinese and Southern Han Chinese. And these are not casual regional differences, the way someone from Northern England differs from someone in the South. These are genetically meaningful distinctions, shaped over thousands of years by different climates, different diets, different diseases, different migratory histories, and different ancient peoples who mixed and melded and disappeared into what we now call a single identity. Genome-wide variations revealed that modern Northern Han derived their ancestry from a Yakut-related population at roughly 25%, and a Shi-related population at around 75%. The genetic mixing that led to the emergence of Han Chinese ethnicity occurred at a very early period, probably in Neolithic times, and this mixing involved an ancient Tibeto-Burman population and a local pre-Sinitic population, which may have been linguistically Altaic. Pause there. Neolithic times. We are talking about the period before written history, before dynasties, before the first emperors, before the Great Wall had even been imagined. The genetic identity of the Han Chinese was being forged and mixed and reshaped tens of thousands of years ago in the forests and river valleys of a world that modern civilization would not recognize. And yet, that ancient mixing, that ancient story, is still alive inside the DNA of over 1 billion people walking the earth today. And this is where everything changes. When scientists began seriously mapping the ancient genetic history of China and cross-referencing it with European populations, they found something that exploded a long-held assumption. Researchers assembled 322 ancient samples that geographically span 3 broadly defined regions, Europe and Anatolia, Central Asia, and Siberia and East Asia, covering a temporal range from approximately 2,300 to 45,000 years before present. Across China, the amount of shared genetic drift was often significantly correlated with latitude, with northern provinces showing higher values than southern provinces. What that means, translated from the clinical language of population genetics into something real, is this. The further north you go in China, the more ancient shared ancestry you find with populations that once roamed Central Asia and Siberia, populations that themselves had connections stretching all the way toward ancient Europe. There is a thread, ancient and fragile, connecting the human story across Eurasia in ways that challenge the idea of clean, separate, and distinct racial categories. The boundaries that we have drawn on maps and in our minds are modern. The DNA is ancient, and the DNA does not respect our borders. But do not think for a moment that this means Chinese and European populations are simply two versions of the same story. Because the differences, the deep, functionally significant biological differences, are just as stunning as the connections. Research has demonstrated greater genetic drift in East Asians than in Europeans. Genetic drift is the process by which certain genetic variants become more or less common in a population, not because of natural selection pushing them toward an advantage, but because of random chance compounded over generations. Greater genetic drift in East Asian populations tells scientists something profound about history. It suggests population bottlenecks, moments in ancient history where the number of people in a group was drastically reduced, perhaps by famine, by climate catastrophe, by war, by disease, and then expanded outward again. Every person alive today who carries East Asian ancestry is, in some molecular sense, descended from a population that survived something that left a permanent mark on the genome. And that mark shows up in ways that are not abstract, not theoretical, biologically real and medically consequential. Consider alcohol. Most people know the cultural observation that many East Asian individuals experience flushing, rapid heartbeat, and intense discomfort when they consume alcohol. It is often treated as a joke, a social quirk. But the reality of what is happening at the genetic level is anything but trivial. Research revealed that long-term farming ethnic groups in East Asia, such as the Han, differed from nomadic populations in the pattern of alcohol-related genetic polymorphism. The agriculture of ancient China, particularly the cultivation of rice, which enabled the production of fermented beverages as far back as the Neolithic period, created a selective pressure on the Han genome. Over thousands of years, the genes that govern how the body processes alcohol were shaped by the demands of an agricultural civilization. In contrast to variants common only in East Asian populations that are associated with greatly reduced enzyme activity and alcohol intolerance, regulatory variants identified in European populations are associated with increased expression of the ALDH2 gene, suggesting adaptation of Europeans to higher alcohol consumption. These are two populations, separated by a continent, whose bodies literally evolved to process one of the most widely consumed substances on Earth in fundamentally different ways, because their ancestors lived differently, farmed differently, and survived differently. This is not biology as an abstract science. This is your body carrying the memories of people who lived 10,000 years ago, and that is only the beginning of what the comparison reveals. Differential susceptibilities to various diseases and corresponding metabolite variations have been documented across diverse ethnic populations, but the genetic determinants of these disparities remain unclear. That sentence, buried inside a 2025 research paper published in the journal Cell Genomics, carries enormous weight. Scientists have known for years that Chinese populations and European populations get sick differently. They respond to drugs differently. They metabolize compounds differently. They carry risk for different diseases in different magnitudes, but the underlying genetic architecture, the precise molecular reasons why, has remained frustratingly elusive. Until recently, a 2025 cross-ancestry analysis conducted by researchers, including Fudan University scientists, performed large-scale genome-wide association studies comparing Chinese and European populations across 171 directly measured metabolites. Metabolites are the chemical byproducts that your body produces as it processes everything from the food you eat to the air you breathe. They are a molecular fingerprint of how your biology is functioning. And what researchers found when they compared the metabolite profiles of Chinese and European populations was not simply quantitative variation. It was qualitative difference. Different genetic variants were driving different biological processes in the two populations, in ways that had direct implications for disease risk, drug response, and the fundamental question of whether medical science built for one population can safely or effectively be applied to another. Think about what that means for medicine as it is currently practiced. If the drugs in your medicine cabinet were designed, tested, and optimized using data from European populations, and the genetic architecture governing how your body responds to those drugs is meaningfully different, then you're not receiving personalized medicine. You are receiving medicine that was personalized for someone else. You are the afterthought in a system that never designed itself with you in mind. By combining genome-wide association studies of cohorts of East Asian and European descents, Researchers identified 3 novel associations that were not statistically significant in either European-based or East Asian-based cohorts considered alone. They also discovered a novel depression association on chromosome 7 in studies conducted with people of East Asian descent that was not detected in depression studies based on United States or United Kingdom datasets. A genetic risk factor for depression, found only when East Asian data was examined, hidden, invisible in the dominant European-focused dataset. Not because it is rare or minor, but because nobody was looking for it in the right place, with the right people, using the right genetic framework. How many other discoveries are buried in that silence? Now let us go deeper, because the story does not stop at metabolites and disease risk. It goes all the way back to Neanderthals. A study published in the Proceedings of the National Academy of Sciences found that while most people of Asian descent are lactose intolerant, some East Asians are not, and they likely have Neanderthals to thank for it. All modern humans carry some Neanderthal DNA, remnants of ancient interbreeding events that occurred tens of thousands of years ago, when our species encountered archaic humans across Eurasia. But what scientists discovered is that the mechanism by which certain East Asians develop the ability to digest lactose, milk sugar, is completely different from the mechanism in Europeans. A haplotype spanning approximately 467 kilobases at a specific genetic locus, has risen to high frequency in East Asians, around 25%, but is entirely absent from Africans and Europeans. This haplotype likely derived from Neanderthals, and has been under positive selection in East Asians. Let that sink in. Europeans and East Asians both, in certain proportions, developed the ability to process lactose in adulthood. But they did it through completely different evolutionary pathways, shaped by completely different ancient genetic inheritances, potentially for completely different reasons. Researchers concluded that variants of the lactase gene may have been selected for different reasons across the world, implying that the selection pressure in European and African populations may not have been solely about drinking milk. This is not merely a footnote in evolutionary biology. This is a window into something extraordinary. It means that 2 populations, living on opposite ends of a continent, facing different climates, different food sources, different pathogens, and different ecological pressures, arrived at similar biological outcomes through entirely different genetic journeys. Convergent evolution, written in the most intimate possible language, the language of human DNA. And what about the immune system? Research has found that the immune systems of northern and southern Han Chinese populations could be subjected to different pressures from viruses and other diseases, with significant genetic differences in regions associated with HIV infection, psoriasis, and psoriatic arthritis. Different regions of China, different pathogen histories, different immune adaptations, all encoded in the genome of a single ethnic group that the outside world often treats as monolithic. If the Han Chinese contain this level of internal immunological variation, the scale of difference between East Asian and European immune systems is not a gentle gradient, it is a canyon. A landmark East Asian methylation study integrating data from 7,719 Han Chinese individuals identified 331,048 methylation markers, including 28,978 that were entirely novel in East Asian populations and not found in European-based datasets. While substantial sharing of methylation patterns was observed between European and East Asian populations, East Asian-specific markers were identified, often driven by genetic variants with very low frequency in Europeans. 28,978 novel markers. Not variations, unknown themes, novel, entirely new. Found only when scientists stopped assuming that European data was sufficient and started looking at East Asian genomes on their own terms. Found only when the question shifted from how do East Asians differ from Europeans to what does East Asian DNA tell us about itself? This is the paradigm shift that is quietly unfolding inside the world's most advanced genetic laboratories, and the implications are enormous. Research has revealed the genetic evolutionary trajectories related to immune and metabolic function in populations that underwent recent polygenic adaptations, in which folate and fatty acid metabolism changes promoted rapid adaptation to the environment in Eastern Eurasia, findings that could promote the development of personalized precision medicine. Personalized precision medicine. That is the phrase that scientists and pharmaceutical companies and governments are now rallying around as the future of healthcare. A world where your treatment is designed for your specific genetic profile, not for the average of a population that doesn't represent you. But here is the brutal irony. The foundation on which that future is being built is still predominantly European. The data gap is real, it is vast, and it is costing lives in ways that never make headlines. In East Asian populations, functional polymorphisms in alcohol metabolizing enzymes, specifically ADH1B and ALDH2, are highly prevalent and significantly affect acetaldehyde metabolism. Based on genotype combinations, individuals can be stratified into 5 alcohol sensitivity groups with differing risks of cirrhosis and liver cancer, and paradoxically, ALDH2 deficiency, which often suppresses alcohol intake due to aversive physical reactions, actually increases the risk of liver and gastrointestinal cancers in those who continue drinking. This is the complexity that gets lost when medicine treats Asian as a demographic checkbox rather than a deeply specific genetic reality. A gene that makes alcohol feel unbearable is not simply a cultural quirk, it is a biological warning system. And when that warning system is ignored, when someone drinks through the discomfort, when the medical advice they receive fails to account for the specific genetic variant in their body, the consequences are not mild. They are malignant. Now pull back, look at the full picture. We have 2 populations, Han Chinese and European, that diverged on separate evolutionary trajectories tens of thousands of years ago. They developed different adaptations to different diseases. They processed alcohol differently. They arrived at lactose tolerance through entirely different ancient genetic inheritance. They carry different methylation patterns that affect how their genes are expressed. They face different disease risks, and those risks are controlled by different genetic variants. They have different immune system histories, shaped by different pathogen landscapes across different continents. And despite all of this, despite this extraordinary biological complexity, global medicine spent the better part of a century building its systems almost entirely on the genetic architecture of one of these groups and calling it universal. The research is now catching up, fast. Studies reporting newly sequenced ancient human genomes from Han Dynasty archaeological sites are being integrated with previously published regional datasets to investigate the genetic legacy of Neolithic populations in the formation of Han Chinese ancestry. And the findings are reshaping our understanding of not just Chinese history, but human prehistory itself. Ancient DNA analysis from frontier regions like the Hehuang area has revealed that most individuals from the studied sites carry predominant Yellow River-related ancestry consistent with Central Plains agricultural populations, while subgroups show affinities to Northeastern Asian and Xianbei-related populations, indicating admixture with northern groups. The Silk Road was not merely a trade route for silk and spices, it was a genetic highway. For centuries, populations flowed along it. Merchants, soldiers, slaves, refugees, nomads, and each wave of movement left biological fingerprints in the genomes of the people it touched. The Han Chinese genome today is not the product of one ancient population remaining in place. It is the product of thousands of years of movement, collision, survival, and adaptation. A palimpsest of human history written in 4 chemical letters, copied imperfectly through 100 generations, and still being read and reread by scientists who are only beginning to understand what it says. And here's the question that none of the official institutions want to answer directly. If the genetic differences between Chinese and European populations are this significant, not cosmetic, not superficial, but functionally, medically, evolutionarily significant, then what is the current global medical system actually doing for the roughly 4 billion people on this planet who carry East Asian or Southeast Asian ancestry? What is the cost, in human health and human life, of decades of genetic research that treated European DNA as the default setting for all of humanity? The full landscape of human genomic diversity and its impact on precision medicine remains an active and urgent field of research, which is the scientific establishment's careful way of admitting that the map is still being drawn, that vast territories of human genetic reality remain unmapped, that the most important question in medicine, why do different people respond so differently to the same diseases and treatments, cannot be answered using data from one corner of a humanity's story. The results shocked the scientists because they were expecting confirmation of what they thought they already knew. They were expecting variation within a familiar framework. What they found instead was that the framework itself was wrong. Not wrong in a minor correctable way, wrong in a foundational way. Wrong in the way that changes what questions you ask next, changes what tools you build, changes what assumptions you carry into the laboratory, and changes, ultimately, what medicine is capable of becoming. The DNA was never lying. The science was never hiding. It was always there, waiting to be read by people willing to ask different questions, willing to look beyond the population that had been convenient to study, and see the full complexity of what it means to be human. 2 populations, separated by continents and millennia, compared at the deepest level of biological truth that science can currently reach. And what the comparison revealed is not a story about difference, and it is not a story about similarity. It is a story about the breathtaking complexity of how humans survived, adapted, migrated, mixed, and thrived across tens of thousands of years, of a planet that never once made survival easy. The question now is not what the DNA says. The question is whether the people who hold power over medicine, policy, and research funding are willing to listen. whether they are willing to rebuild systems that were designed for some of humanity around the needs of all of it, whether the shock of the results will become the fuel for genuine change, or whether it will be quietly filed away while the old assumptions continue to quietly shape the drugs that are designed, the studies that are funded, and the lives that are saved or lost in the gap between what science knows and what medicine actually does. The results shocked everyone. But the real question, the one that should keep you awake, is what happens next. Because the data exists, the evidence is mounting, the gap is visible to anyone willing to look directly at it. And whether that changes anything at all is no longer a scientific question. It is a political one. And that is where the story gets the most dangerous of all.